Welcome to the final part of Module 4. Okay, in this module, you were introduced to the 4 different approaches within genetics. We are going to rely on all of these different types of genetic approaches over the course of this semester. But our first line of investigation that we've done in this module has relied on population genetics. So we got to see some of the earliest research within human population genetics through the mitochondrial DNA research of Conn, Wilson, and Stoneking from 1987, right here, UC Berkeley. We expanded that to include subsequent mitochondrial DNA research and Y-DNA evidence. And now in this video, we expand our population genetics investigation to include people from a long time ago, so ancient DNA. We will also bring in genomic sequence data. So instead of relying on just short regions of DNA sequence, we will consider sequence data that better reflects the entire genome. Now this is going to be a long one because what I decided to do for this one is to use the lecture that I would have presented in class. And I thought long and hard about how to divide this up into little pieces, but there's a through line for the story, and I didn't want to break it up, and I figured, you know what, we can do it. Let's stick with this. We can do this long video. Feel free to take a break. You can pause me at any time. I can give you little hints about when it might be good to pause and come back and join me in a little bit. But basically, pretend you are in Duino, and we are hanging out there together on our long journey today for our regular type of class meeting. On to the next slide. So we left off our population genetic study here with the mitochondrial DNA and the Y-DNA evidence converging on these somewhat different coalescence dates. Now these differ because mitochondrial DNA and Y-DNA tell us about different parts of our populational history. Men and women often experience life differently, not just from our personal experiences, but also in terms of their reproductive lives. So the Module 4 activity was aimed at getting you thinking about how the dramatic cultural changes that happened between 5,000 and 7,000 years ago influenced human genetic variation. I want to refresh your memory of this figure and these data. This is from the Conn, Wilson, and Stoneking study. This is the histogram showing the average number of sequence differences between all possible pairwise comparisons in the data set of those 147 mitochondrial DNA sequences from women who lived in modern times. The average number of differences is somewhere around 7 or 8. Keep this in mind, as I'm going to show you another data set that is presented this exact same way. But this data set is going to come from a fossil. So 1997, I was a graduate student at the time. I had just started my PhD program, and I was at Penn State University, and what an exciting time it was. Actually, it's almost always exciting times in science, but for me, bright-eyed, bushy-tailed PhD student, this was amazing because this was the publication of the first DNA that had been extracted from a Neanderthal fossil. They actually took a saw to the type specimen of Homo neanderthalensis. These are the fossils that were found in 1897 in Neander Valley. Okay. Look at that. As a paleontologist, it makes my heart bleed. Anyway. They sawed this open, they ground down the bone, and they were able to extract, amazingly enough, mitochondrial DNA. This was a huge thing at the time because it was technologically really difficult. And the lab that did this in Germany at the Max Planck Institute in Leipzig, they were the lead on the study, but there was a control that was done to see if they could replicate the extraction of the mitochondrial DNA and get the same sequence. And that control study was actually done at Penn State in this tiny little closet of a lab that was in the same building where I spent every waking minute of my life, it seemed like, in graduate school. So I was a part of all of this, at least in terms of proximity. So really exciting when the study came out and we actually got to see what the data told us. So here's what they found. So here you're looking at, this is the same kind of histogram you were just looking at. So the Neanderthal to human sequence comparisons show, anyway, so they lined up the human and human, and you can see over on the left-hand side of the graph, this is the same, these are different data, but it's the same basic result as we saw in the Conn-Wilson and Stone-King study. And notice that, just like in Conn-Wilson and Stone-King, that the average number of differences between the human-to-human pairwise comparisons is somewhere around 7 to 8. Now, look just next to that, you have human to Neanderthal. So it's just the one Neanderthal sequence, but they compared it to every one of those human sequences. And so they could then get an average number of differences between the one Neanderthal and all of those humans. And on average, there are about 25 or 26 differences between the Neanderthal and every possible human that was compared to it in this data set. And essentially, those distributions touch, but there's not overlap. They also looked at human to chimp mitochondrial DNA as well and aligned those sequences. So you can see the average number of differences is over on that right-hand side of the graph, somewhere around 55. So the Neanderthal to human difference, it's about 4 times as many differences as what you see between any two humans that are being compared. So that then indicates that that most recent common ancestor in the mitochondrial DNA sequence for humans and Neanderthals is much older than it is for humans to humans. So the Neanderthal human most recent common ancestor is much farther back in time. So the geneticists at the time, remember, I'm sitting at Penn State. These are people that I see all the time. The geneticists were absolutely confident that these data indicated that Neanderthals and humans had no genetic intermixing. Neanderthals were this side branch. They'd moved into peninsular Europe. They were doing their own thing. And for some reason, humans and Neanderthals had no genetic admixture going on. They were not swapping around their DNA. They were adamant. Absolutely. Many of the paleontologists were like, yeah, I don't know. I think I can see in the bones, it kind of looks like you have, some features that look more Neanderthal-like. And anyway, so there was a huge debate. And the geneticists were absolutely 100% sure. Nope, our data are always right. But it kind of, as an evolutionary biologist, raises this interesting question, why would there be no genetic swapping? Why no mitochondrial DNA contribution? Maybe only male Neanderthals mated with Homo sapiens? Maybe Neanderthal mitochondrial DNA was selected against? It's really hard to know. And so people were trying to rack their brains to come up with these really elaborate behavioral explanations as to why Neanderthals and humans appear not to have had sex. So at least 15 Neanderthal mitochondrial DNA sequences have been developed since that study. Here's a map that shows you where they are all from. And here are some of the sequences that are lined up. So at the very top of this, so actually down on the left-hand side, you can see a list of the different specimens. The CRS at the top, that refers to the Cambridge reference sequence. And this is a modern human standard that is used in a lot of these studies, Cambridge reference sequence. And then you have positions all across all of those numbers. The 16037, for example, those are specific base pair sites in the mitochondrial genome. So all the different specimens are below the Cambridge reference sequence. So you have humans, and then all of the other ones below that are Neanderthals. And draw your eye to the color, the pattern of the colors, because that's showing you that there really is a Neanderthal cluster of these base pair sequences, and it's distinct from humans. So there is this clade, this mitochondrial DNA clade of Neanderthals, and the more and more Neanderthals that were sequenced, the more and more this became evident that you had this mitochondrial DNA cluster that characterized Neanderthals. So all of that mitochondrial DNA data could be then used to figure out a date for that coalescence. So again, using the molecular clock and estimating when that last common ancestor, a most recent common ancestor, would have existed. So it's estimated to have been about 4 times older than the most recent common ancestor for all human mitochondrial DNA variation, which would put that Neanderthal human common ancestor back at somewhere around 600,000 years ago. Well, time went by, and we not only had mitochondrial DNA, but we also started to get some nuclear DNA, fragments of nuclear DNA, and soon I'll tell you about whole genome data. But all of those pieces coming out of the nuclear DNA started to suggest that we had a most recent common ancestor between Neanderthals and humans about 270,000 years ago to 440,000 years ago. Now, we would expect that there would actually be a range of dates for the most recent common ancestor, as alleles recombine through each episode of meiosis, like cards being reshuffled in the deck, gradually changing in frequency over time. So, this wasn't that much of a shock. This was kind of expected, and it's also pretty interesting. But, the other thing I want to point out is that this is always going to be an inexact science, because we're talking about people, and a person is just a current configuration of alleles, and that because of meiosis, because of recombination, it's always being reshuffled, and so you're not going to have this real linear relationship between a whole bunch of variants of a gene. They get reshuffled, and so every one of those different genes is going to have a different evolutionary history. They're not going to all be the same. And for example, draw your attention to CD209. This is a gene that's involved in innate immunity, and of all the variation that we see in humans today, that most recent common ancestor is 2.8 million years ago. So it doesn't have to be even a modern human. It can go way back in time. But all of them will have a common ancestor, a most recent common ancestor, and there is no reason to expect them to all be at the same time. In fact, they shouldn't be. Now, this most recent common ancestor, honestly, I think it can be a little bit confusing of a concept, especially as more and more genomic studies have started to be published. Because all of these studies, they get a huge amount of media attention. And with that media attention, that message just becomes increasingly simplified to the point where it's just really confusing from my perspective. Now, in 2010, a decade ago, the first draft of the Neanderthal nuclear genome sequence was published to quite a bit of fanfare. Well, not surprisingly. Neanderthals, genomes, So the headline, of course, was that non-Africans are 1.4% Neanderthal. The question had finally been answered. Did humans and Neanderthals have sex? Now remember, just 13 years before this, the geneticists were like, no, they did not. Okay, but now, with some genome data, they're like, yes, we had sex with them after all. Okay, no geneticist is ever going to actually be like, yeah, we were wrong. But anyway, they weren't really wrong. They were working with a really, really small data set before, and being a bit too overly confident with working with such a small data set of just mitochondrial DNA. So when they had the much larger data set of all of the nuclear DNA, perhaps not surprisingly, the story changed. Our interpretations, the data, the evidence, it changed. So those Neanderthals that were hanging out in Europe, and humans came up, ran into them, and we might not have shared all that much DNA, but we did swap at least some. Perhaps the interpretation was maybe the Neanderthal men were, some Homo sapien women just couldn't resist them, or perhaps those interactions were brutal. Whatever the case, the way it was presented at the time that this study came out was that the people who live in Eurasia could start to claim some special affinity to Neanderthals because Neanderthals were there, and they had moved up, had a little bit of sex with Neanderthals, and admixed. Okay, before you take that as established fact, I'd like to dive into that study just a little bit more. Now, the scientists who conducted this study, the 2010 study, they looked at the genomes of 5 people. Whole genomes. They were a lot harder to access 10 years ago. So the scientists, they wanted to have geographic coverage. So they had the genome of a French person, one French person to represent all of Europe. They had a Han Chinese person to represent all of Asia. They had a person from Papua New Guinea represent all of Southeast Asia. And they had all of Africa being represented by 2 people, one person from Nigeria in Western Africa of Yoruban ancestry, forgive me for mispronouncing that, and another person from the San in South Africa. That was it. 5 people. Oh, and the Neanderthal. Now, what the authors found through their analysis is that for many regions of the genome, that the variation within current humans is old enough to include Neanderthals. So, that's a quote out of their paper. Now, let me have you look at this figure over on the right-hand side of your screen. So, you can see here those 5 humans, the San, Yoruba, PNG, Han Chinese, and French. And then they had data from Neanderthals. And actually, they looked at 2 individuals. So they did that coalescence. And the thing is you're looking at a huge amount of data if you're looking at billions of different base pairs. But what they found is that there is some closer affinity, that when you're drawing those coalescences, when you're doing sort of walking back the variation to find those founder events, that there is quite a bit of that. There's a bit of that genetic variation that does have Neanderthals more closely having a closer affinity with people those 3 people who are not African. But that actually the whole African genetic, if you look at all of the rest of the genome, that there's actually a lot of genetic variation and it can easily absorb the Neanderthals. So there's a couple different ways that you can explain this kind of relationship. So one of them is that the humans who lived a really long time ago, they were genetically diverse over a big geographic region. So there was some population structure, and that then the Neanderthals and the people who currently live in Eurasia, that both of those groups had their ancestry within the same subset of the African population. An alternative explanation is that humans and Neanderthals had sex, despite having been genetically distinct for some time. So that alternative explanation is the one where the Neanderthals were there, they had been genetically isolated for a while, and then humans moved into the region and, despite their differences, had a little bit of sex. And so there was a little bit of admixture, and that admixture was shared across all of the people who, of those populations who are not African. So of these 2 possible explanations, which do you think the New York Times preferred? And which do you think was therefore the chosen interpretation of the authors? Yeah, yeah. Neanderthal sex is going to win it every time. Okay, let's let that one sit for a few minutes. And I want to zoom out from Europe. I'm sitting on a rolly chair. Was that cool? Anyway, we're going to zoom out from Europe and think about geography on this wider scale. Now, I want us to take a look at some fossil remains that were recovered from a cave way over near Kazakhstan and Mongolia. Denisova Cave, way over to the right. So 2010 was kind of a wild year in ancient DNA studies. So first we had that Neanderthal genome, and then we had the publication of ancient DNA being extracted from this tiny little fragment of a finger bone. See it? It's the little brown plop on that person's pinky. That little bone is thought to be about 50,000 years old. So Denisova has made quite a splash. Let me add a little more to this, though, because the ancient DNA studies just really started to accelerate at this point. So a couple of years later, we have mitochondrial DNA that was extracted from fossils you have actually already seen before. Those are the bones from the Cimo de los Huesos. So here's another map that has the Denisova, has a bunch of other of the ancient genomes that had been extracted. So Cimo de los Huesos, these are the 400,000-year-old cave deposits, the pit of the bones. These are the ones that are not Neanderthal because they're older, but they have some features on their crania that make them look a little bit more like Neanderthals than other Homo erectus. So they're kind of in that gray zone. So here's a phylogenetic tree of mitochondrial DNA relationships that include Cimo de los Huesos, the Denisovan sequence, the Neanderthal sequences, and then living people. Whoa. So it's looking like quite a lot of variation there. Let's add a little more to this so we can add some more data into this. So in 2014, we had more Neanderthal and Denisovan data analyzed. Here they looked at 6 archaic genomes, so they're looking at nuclear DNA now, and 25 modern genomes. So this is more than just the mitochondrial DNA sequence you were just looking at. And so here they have a cluster of Neanderthals, they have 2 Denisovan individuals, and then they have their modern humans. I want to draw your attention to those little arrows that are kind of going all over the place, that are connecting Neanderthals to Denisovans, Denisovans to modern humans, some potential unknown hominid to Denisovan, Neanderthals to modern humans. The story's starting to get kind of complicated, like how to interpret all of this. So the story gets even better because in 2018, we even add a love child to the whole thing. So they found evidence of, the geneticists found evidence of a Neanderthal and Denisovan love child. Okay, so in this analysis, they had their genome sequences of 4 archaic individuals, and from those archaic genomes, they estimated these last common ancestors for them and the relationships between those. And so you can see those 4 individuals. You have Denisova 3 in the lower right, and then it's beside Denisova 11, from which they interpreted the dad being Neanderthal and the mom, the dad being Denisovan and the mom being Neanderthal, merged together. And then they had Vindhya and some other Neanderthals there, and then they had the Altai Neanderthal as well. So this is what they reconstructed from those 4 ancient genomes. And you have time there on the x-axis, on the y-axis over on the left-hand side of the screen. So I want to draw your attention to kind of thinking about the timing of all of this. So they're estimating that you have this Neanderthal and Denisovan coalescence somewhere between 390 and 450,000 years ago. Okay, let me add to this a little bit. Now, you've seen this before. These are the models for the origins of Homo sapiens, the multi-regionalism, and the replacement. And we've tested these using the mitochondrial DNA and the Y-DNA evidence, and basically showed that multi-regionalism, when you look at those very restricted data points for mitochondrial and Y-DNA, that you basically reject multi-regionalism. It looks much more like replacement. Now, I've taught this class for many, many years, and so as I had these up, and then as the ancient genomes, ancient DNA evidence started to be published, I worked to incorporate all of that. And so I liked my models because, it makes it kind of clear, it's easy to understand. And so I kept trying to adjust my model to incorporate the new evidence that we were finding. And so then I came up with this "it isn't so simple" model that you can see here on the right. And so I had these other parts of Africa, and then I had that closer affinity between non-Africans and the archaics. And anyway, it got really complicated. So basically, a couple of years ago, I was like, this is really getting ridiculous. So maybe it's time to stop thinking about this ancestry in terms of these specific lineages and how these lineages relate to each other. And so what I decided to do was to step back So we're going to step back again and think about geography again. Now, we have evidence that there were people, whatever you want to call them, Neanderthals, people, archaic people, they were living all across this vast geographic range. Like, 700, 600,000 years ago through to today. That's a really big region. Now, remember back in Module 1, we also talked about the tyranny of the present. And one of the big things I wanted you to keep in mind is when we're talking about such vast timescales, we also need to think about how climate has changed. So here is a figure, a graph showing you how temperature has changed over time. And modern day is over on the far left. And it goes all the way back to 650,000 years ago on the right-hand side. And you can see temperature oscillates up and down. Warmer times on the top half of the graph, colder times on the bottom half of the graph. Below that, so these are showing you, of course, the ice ages down below and the interglacials above. Now below that, I plotted on the duration of when different fossils, different taxonomic groups have existed. So we have our Denisovan, which actually we don't know anything about what Denisovans look like. All we know is that tiny little finger fragment, there's a tooth fragment, there's little bits, but we don't actually know much about what they look like. So we have the Denisovan DNA population somewhere about 50,000 years ago. You have Neanderthals from about 130,000 years ago up to about 30,000 years ago. Homo sapiens that show up kind of on the scene depending on where in the world you're asking your question about, maybe about 40,000 years ago. And then we have further back in time, the Cima de los Huesos fossils from the cave in northern Spain, dating to around 400,000 years ago. And then I included the Neanderthal Denisovan last common ancestor and the estimates for the Neanderthal human last common ancestor. So notice you have Cima. Notice that you have those estimated LCAs around when you've got a pretty big interglacial, a pretty big warm spell, and then another ice age, and then into another warm spell. So what that means is that over that time period, you had glaciers expanding and contracting and then expanding again. Remember those maps that we looked at? It dramatically changes what the landscape is like and how animals, including hominids, could have been making a living on that landscape. Something else to think about. A little more geography. I've tilted the Earth up a little bit here because we've really focused our conversation so far on Europe and, sort of that more western part of Asia. But there's this whole other continent down there that we've kind of ignored. Yeah, Africa. Let's take a look at what's going on in Africa with the genetic variation there. There's a great study that came out a couple years ago that synthesized a lot of the work that was being done in African genetic variation. So, genomic variation in living Africans. So again, looking at modern people. In this study by Schlubusch and Jacobson, I think that's how you pronounce his name, they sampled 15 individuals from 93 different populations. So each population had multiple individuals in it. So that's a total of 1,395 individuals in their study. And this gives you a sense of the geographic regions that were covered. So they had non-Africans, West Africans, Central African hunter-gatherers, East Africans, or Eastern Africans, and the Khoisan. Now I want to introduce you to another way to look at genetic data, genomic data actually. So this that you're looking at down here, this band, it looks like a ribbon with colors on it, this is called an admixture plot. What the scientists have done is they took the genetic variation and then they color-coded it based on which population that DNA sequence is most common to. So remember when we were looking at haplogroups with mitochondrial DNA? It's that same basic concept, but it's applied to a much larger sample of genomic data, and then they color code it. And so they put, for each of those, you can see these names down at the bottom, those are the different populations. And so they have then a little strip for each of the individuals in each of those populations, and they color code their genome based on what proportion of their genome has an affinity with which ancestry group that they're interested in looking at. So what you're seeing then is this variation in the colors. And so you start with a color coding over on the left that matches with the non-African populations, and then you increasingly see representation in those genomes of East African ancestry, and then a little bit of the Central African hunter-gatherers and the West Africans, a little blurb from the Khoisan, so I'm moving from left to right, and then a whole lot more West African, but you still have quite a bit of the East Africans and the non-Africans, and then an increase in the Central African hunter-gatherers as we continue to move to the right, and then a whole lot more Central African hunter-gatherers, a smaller amount of West Africans, and then you gradually move into having more and more representation from the Khoisan. So the big thing here is wow, there aren't these really clear delineations between those 5 different populations, those 5 different major geographic regions that the authors have imposed on this data set. Kind of interesting that there's actually quite a lot of mixture. The other thing that these researchers did is they calculated time. So they use this molecular clock approach, they used the coalescence theory, and they reconstructed what the population history is for all of this genetic variation. So now you still have your admixture plot at the bottom, but now I've added on a figure from their paper that gives you a sense of the time depth of when they have coalescence, when they have those most recent common ancestors for the genetic variation that you see across those populations. So time is on the left side. Now, present day is towards the bottom of that sort of upside down phylogeny, and then you're moving back in time as you go up that y-axis all the way up to 600,000 years ago. And the authors have included a line that shows you where the Neanderthals and Denisovans are thought to have sort of diverged off of that. So check this out. You've got some coalescence that happens, both of the major clades of this coalesce somewhere around 250 to 350,000 years ago. There's some major structure in the genetic variation in Africa. So people are a mixture, depending on where you are in Africa, they're a mixture of these different populations, but then there's a lot of time depth between those populations. This indicates that people have been all over Africa for a really long time. It's a very wide, very big population with a lot of genetic structure, with a lot of structure to the population because of how widespread it has been geographically and how long we have been there. I want to add some comparison here. Now, the authors of this paper had indicated that Neanderthal Denisovan, but I actually added on in yellow what that estimate is from some other papers. So the Neanderthal human last common ancestor being somewhere around 270 to 440,000 years ago. So it's on the graph, and notice it's actually not that much deeper than where you see those 2 major clades of the African genetic variation coming together. Let me add a little more perspective. We're going to do a compare and contrast here now. So the figure we were just looking at is on the right, and I put over to the left the figure that's actually one we were just looking at when they found this is from the paper about the love child, the Denisovan and Neanderthal love child, and it's super small because it's such a small, it's 4 individuals, and so the graph doesn't actually cover all that much time. So I lined them up so that you can get a sense of scale. So you have your massive African genetic variation study on the right, and then your 4 individuals from Eurasia on the left. And I lined up the 100,000 years ago, so they're about right. You can pause me if you want and try to make it bigger and take a look at this. And notice at 100,000 years ago that you have Denisovans and you have Neanderthals seeming to have a love child at the same time, that basically you've got kind of African genetic architecture being kind of what it is now. You got, definitely some other stuff that happens, some massive admixture going on with, once you get into the spread of agriculture, which is why the green kind of smears all across, because there's a massive migration of agriculturalists about 3,000 years ago. But all of a sudden, this big hoopla about Denisovans and about Neanderthals, it's really just very comfortably subsumed within that same kind of scope of variation that we see when we get ourselves out of Europe and Asia and Western Asia, when we start to look at Africa, when we start to look at really where the major amount of human variation and the major amount of human evolution has occurred. This is just a little side story. So this is really hopefully starting to get you thinking about human variation in a different way. We had that idea about, there were lineages and they migrated out of Africa and then they stayed there and then they were distinct. Or did they have some migration? Or were they completely replaced by another? Maybe it didn't actually happen like that at all. Maybe we had all of these hominids kind of moved around the world. And then they were kind of there, but there were people moving around a lot after that. Maybe it's not really a clean story of just these lineages, these population lineages. There's another study that came out a couple years ago that I just love. I think it just really solidified a lot of my current thinking about human genetic variation, and I'm going to share it with you now. This was published in 2017 by Baker et al., Baker and colleagues. Human ancestry correlates with language and reveals that race is not an objective genomic classifier. What they did, so in this study, they had 5,966 individuals, genome data for close to 6,000 individuals, representing 282 global samples, representing 30 primary language families. This is a massive study. So a lot of people from a lot of different populations and different ancestries and different language groups. Now what you're seeing, these pretty colorful strips, those are the admixture plots, just like the one you saw before, but they're standing on end this time. So each of those names, the little tiny names that are basically impossible to read, is a different population. Those are the 282 populations that are included in this analysis. The really big thing to notice, let me blow this up for you so you can see it a little better. The really big thing to notice is that there are multiple colors no matter where you look. They're all kind of mixed up a bit. I mean, there is some pattern to it. So, for example, South Asia is the one over on the left, and you have the purple and the tan color, and then the darker blue, and some white. There's these strips in there, but those are all of those different populations, all of those different groups. They have a lot of different ancestry in them, and then the same thing in Africa, and then Europe, Caucasus, and the Middle East. Again, there aren't these clear chunks of ancestry. There aren't these clear groupings of genetic variation. It's actually a blur. They're intermixed. And the same thing for Oceania, Asia, and the Americas, over on the right-hand side. So here are the conclusions from this Baker et al. Study. So again, they looked at close to 6,000 individuals from 282 samples, representing 30 primary language families. The best evidence that they found is that, so when you take all of those genetic data, now let me back up one second here. Actually, just pause for that. So ignore this for just a second. One of the things that the study did that was really different from prior studies is that they didn't tell the analytical method, they didn't tell the analysis how many clusters to find in the genetic variation. So, for example, in that earlier study looking at African genetic variation, they had those different clusters that were color-coded. They told the analysis to identify those clusters. They told them how many, told the analysis was told how many clusters to find. In the Baker et al. Paper, they let that be unconstrained. They let the analysis figure out, oh, how many clusters comfortably organize these genetic data. And when they did that, they found that there were 21 ancestry groups. So this gets to this second point here. So the best evidence supports 21 ancestries that delineate genetic structure of present-day human populations. So when you don't impose the populations, the number of categories that genetic variation can be divided up into, their analysis found that it most comfortably can be described by 21 different categories, 21 different ancestry groups. And these correspond to these subcontinental regions. The other thing they found, as you can easily see in the pattern of color in the admixture plots, is that the vast majority of individuals, and in fact it was 97.3%, the vast majority of individuals have mixed ancestry. There's evidence of multiple ancestries, of at least 6 ancestries in 96.8% of the people that they looked at. 97% of the people in their study had 6 ancestry populations represented in their genomes. 6. So rather than thinking of human evolution as lineages that gave rise to modern-day populations, it's more accurate to think of human evolution as numerous, intertwining, dynamic, and ephemeral populations. Remember, you've got climate changing. The ice sheets are changing, and they're changing the way the landscape can be used. The climate's changing. People are moving around the landscape in response to these major climatic events. And as they move around, they're changing which populations are intermixing with other populations. Much more accurate to think of human evolution as these numerous, intertwining, dynamic, and ephemeral populations. The populations today, they're just the current configuration. Evolution is a dynamic process. And it has been dynamic for the whole time that we've been here. So our genetic variation is suggesting that a tree is not actually a good metaphor for the history of human populations. Rather, the better analogy is coral, because you have these populations that might be separate at one point in time, but then things change and they merge together, and then they might diverge and then merge with other populations. And so basically you have human populations diverging and coalescing repeatedly through time. And so it's just this much more complicated intertwining, coming apart, forming back together, coming apart and forming back together in response to cultural change, in response to technological innovation, in response to climate change. This is what recent human evolution looks like. So for me, this was like, whoa, a total mind blow. It's just a really different way to think about the variation in humanity, in who we are. So instead of seeing people as a representative of one particular ancestry group, we should actually be thinking about ourselves and about each other as actually carrying with us representations of probably at least 6 different ancestry groups in the past. And so with that, I'm going to let you be for the end of this module, and we will pick everything up in the module 5, looking at Speciation. So now that we have a sense of this genetic variation, let's compare that genetic variation to how much within humans, let's compare that genetic variation to other apes. Do we have a lot of genetic variation or a little bit of genetic variation? Let's find out. I'll see you on the other side of the quiz. Bye.